Hello, and welcome to the Tetra Science SIMPLO video series, where we'll explain how the Tetra Scientific Data Cloud can help you visualize chromatography data. Members of chromatography labs, including lab managers, technicians, and analysts, are often faced with issues that arise due to a lack of centralized and harmonized data. The Tetra Scientific Data Cloud addresses these issues by collecting your chromatography data into a single centralized location and engineering it into Tetra data. This contextualizes raw instrument output with sample, method, and instrument metadata and harmonizes the data into a vendor-agnostic format that is analytics and AI ready. With a SIMPLO dashboard powered by the Tetra Scientific Data Cloud, all of your chromatography data can be visualized, compared, and analyzed in a matter of minutes. In just a moment, you'll see examples of these visualizations that demonstrate how our SIMPLO dashboards can lead to faster and better scientific and business outcomes, such as reduced out-of-spec results, more accurate shelf life prediction, increased lab productivity, and lastly, increased compliance and reduced risk. In this short video, we'll show how the Tetra Scientific Data Cloud enables analysis and aggregation of stability data via the SIMPLO dashboard. Pharmaceutical products can undergo chemical and physical degradation when exposed to environmental stresses like heat, cold, light, and moisture. These stresses can impact the safety, quality, performance, and efficacy of the product. Hence, assessing the stability of both the drug product and drug substance in various storage conditions is a crucial activity in the development and commercial manufacturing of pharmaceuticals. It's common to perform stability studies as fit for purpose. For example, in the early development phase, stability studies are typically performed on a handful of batches in a fairly short period of time, sometimes no more than 3 months, with the primary purpose of assessing early formulations for animal and first-in-human studies. During late-stage development, regulatory agencies mandate the collection of stability data for pre-approval. This data should encompass at least 3 primary batches of the drug substance and 3 batches for every proposed strength of the drug product. Post-approval stability studies must be performed on commercially manufactured batches. Most importantly, the results of these studies must be included in annual reports submitted to regulators. Aggregating and trending data such as potency and impurity levels from chromatography results are primary components of these periodic reports. As you can imagine, following multiple stability studies and aggregating their data becomes more challenging as drug development progresses and transitions into commercialization. Results for active stability studies may be recorded manually in spreadsheets as they are generated, which is prone to transcription errors. This manual process also strips out some of the context that may be needed for an in-depth analysis of the data. This dashboard addresses the challenges of aggregating chromatography data from a stability study, made possible by the harmonized data available in the Tetra Scientific Data Cloud. The example dashboard here shows a set of analyses performed over the course of a few months. It's important to have metadata such as time points, temperature, and humidity to properly aggregate the data. By convention, the metadata is encoded in the sample names. This makes it easier for us to parse those parameters and use them for filters in the visualization. To focus the chart on the active ingredient, simply select the component from the analyte filter. In this example, we'll select the analyte called drug_API. By clicking a sample name that is annotated with lot and storage conditions, you can retrieve the dataset for all completed pull points. A trend line will also appear to help you visualize the overall trend of a quality attribute, in this case, area %, with respect to the product specification. Clicking the data points in the chart will show relevant details, including the user who generated the data, when it was obtained, and other information. Additional metadata about the specific data point, such as the sample set and result set, can also be surfaced if needed. You can, of course, explore the stability trend for the other conditions and combine the data. Selecting one of the impurity analytes will reveal details about the impurity and potentially show the trend across multiple conditions. As you can see, The SIMPLO dashboard streamlines analysis of stability data, enabling formulators and development scientists to shift their focus from aggregating data to deriving insights from the data. That concludes this demonstration of the Tetra Science SIMPLO dashboards. Please email demo@tetrascience.com if you have any questions or would like to schedule a live demonstration. Thanks for watching.